Alright, hello everyone to another science stream. You know, the biggest thing that delays these is just trying to find good enough music to play. Let's turn that down just a teeny tiny bit. But yeah, we got several things to go through today. I had to dig a little, but you know, I wanted to find something that wasn't just climate changes happening right now and it's real bad. AI is happening right now and it's pretty weird. That's basically it. That's kind of more or less it, or like, you know, like here's the newest Alzheimer's drugs or something. You know, something a little new, something we haven't exactly heard in the past like couple of years or months. But yeah, this first one, I'm pretty excited about this. It's probably gonna be a pretty technically heavy one, but this is about. So with the new biomapping technique, mouse enters glow with exquisite details. So this is basically showing that this is a mouse and it's showing the fluorescence all throughout its body with the nerves and everything. And I think that's cool as hell. And honestly can have extremely great applications in the field. But yeah, you'll see fluorescent antibodies light up the nervous system of a dead transparent mouse lying on its back with its head to the left. Yeah, that's its head. It's this little front paws. Colors show how deep nerve cells are in the animal, from blue, closest to the camera, to pink to yellow, farthest away. Yeah, more science. But yeah, so essentially with how a lot of microfluorescence works. I'm good. How are you? How are you doing today? I hope you're ready to learn. But a lot of fluorescence works with using antibodies, and it's a pretty interesting, fun, like process, I guess. Because you basically learn that we can put fluorescent dyes on top of antibodies and then have those antibodies go to, here, let me see if I can pull something up. Can have those antibodies go to specific tissues or cells that we want them to go to. Oh no. Let's see that I don't know how to spell. Let's see if I can get a picture that will kind of describe. Let's see. None of these are really demonstrating the technique of microfluorescence. Let's see. Antibodies. Okay, so this is more or less it. You got your protein. You got whatever it is you want to look at. You got your primary antibody. Let's see if I can make this bigger. You know what let's do. You got your like primary antibody. You put that sucker on. It sticks to all the proteins that you want because it's specific to that protein. You know, made for that and everything. But that's not enough. So you have your secondary antibody, which will have the fluorescent dye on top of it. And then that actually will attach to the primary antibody. I don't know the exact details, but I think it's basically along the lines of the primary antibody can't have the dye because it would probably mess with the structure of the antibody to be able to get to the protein. So we have secondary antibodies for that, and there are some you know, that will have the dye on them, but more than likely you'll do your primary antibody, your secondary antibody, and then you can look into the microscope and be like, ah, there's your green and there's your blue, you know, to represent whatever it is that you're actually looking at. But this is doing it throughout the entire mouse and through all its nerves, which is pretty cool. I do have to say I'm curious about the distance, like the blue being closest to the camera and the pink and yellow being farther away, because I'm curious about that. I'm also curious about what it means by transparent mouse, if it's actually something. It's sort of the same concept. That's sort of how you have the glowing mice, except that's a little bit different. This was just simply adding a fluorescent protein into their genome, and then that makes them all glowy, you know, sort of similar to bioluminescence. But that was, well, I guess back then it was a pretty new concept, but now we just know that we can make that happen. And, you know, make a bioluminescence exogenously. Yeah, I wonder if it's I want to see what it means by transparent mouse. Like, is that actually a type of mice you can get? Doesn't look like it. Looks like it's just saying that. What the hell? Oh shit. Okay. Closed one of the other things I did not want to close. Should do it with humans. We would probably just have to do it when they're babies though, which is the unfortunate, well, not unfortunate, but just, I don't think we should really start shoving green fluorescent proteins into babies. Yeah, it's all gotta start young. Alright, so let's see. Mouse's entire nervous system lights up in psychedelic hues. Clumps of immune cells attacking tumors give off a ghastly glow. Yeah, your kid will be very special, for sure. all the other kids will want to bring him to the party and be like, hey, can we bring Jeffrey to the party? He'll be a really nice decoration for everyone to see. Let's see. Vessels that make up the body's sewer system flare brightly. It makes it sound like it's talking about like intestines or something. These and other images are shining a new light on the inner workings of mice thanks to a new technique that makes whole body imaging cheaper and faster. Researchers report July 10th in Nature Biotechnology. Ah, okay. Using standard antibodies. Interesting. See-through mice are nothing new. So these actually are just, ugh. Are these things alive? These actually are just see-through mice. I thought it was just like a dead mouse or something. The clear alright, hold on. We gotta get into this before we go into the other thing. Clear lump looks like some bizarre translucent gummy candy that might have once been pina colada flavored. What a disgusting sentence to say about a translucent animal. What an absolutely bizarre thing to say. This is something you definitely don't want to eat. The see-through blob was once a mouse. Okay, so once a mouse? In the August 14th cell, okay, so this is 2014, at Caltech, detail a difficult series of methods required to make small animals, such as mice and rats, completely translucent. Scientists have been trying to clear tissue for better observation since the 1800s, and as with all science, the new techniques build on many previous experiments done in a variety of labs. I mean, fair, you know, like, being able to see through the skin would make things a lot easier for observation and whatnot. Technique makes it possible to capture images both beautiful and gross, and the procedure will teach scientists more about anatomy than ever before. If you want to render an animal transparent, I'm curious I mean, I guess we'll see, but I'm curious how they limit that to just the skin. So, if you want to render... Do you want to render an animal transparent? Well, here's how you do it. You first have to overcome a solid problem, lipids. This group includes molecules essential to life, such as fats, cholesterols, waxes, and steroids. Lipids form the membranes that surround our cells, the hormones that make us grow and reproduce, and much, much more. The lipids have a problem. You can't see through them. To be or not to be? Hold on. Signature pheromones in male mouse urine, counter-marking behavior. Okay. Bethany, I like the way you write things because I see this sentence, but for mice there is no pin, there is only P. Bethany, keep up your job. You're good at it. So let's see. Okay, so to render an organism transparent, you need to remove the lipids. Ben Yang and colleagues in Vivian Gardinaru's lab at Caltech use detergents to dissolve the lipids. Alright. You can go smoke some herbs. But I probably will continue. But you can always go back and catch up later. I'll catch you up when you're back though, for sure. So let's see. Okay, use detergents to dissolve the lipids. Yep, it's pretty standard. Detergents are pretty good at dissolving and breaking apart like cells and things. Okay. You know, but don't worry. I'll catch you up on anything that you don't catch. Let's see. The technique is based on clarity, a method that Gradinaru helped to develop in Carl Deseroth's lab at Stanford. Scientists there rendered a mouse brain transparent using clarity. Oh, let's look at that. New technique of see-through view into mouse brains. Peekaboo brain. Alright, hold on. I want to try something. Oh, never mind. I guess, I don't have the extensions on this profile. There's an extension I got recently that will hopefully allow me to get rid of, like, hey, please sign up, or like, please pay for our website or whatever. And I can just keep going rather than being stopped by that. But that's alright. We don't need that. Okay, Gradinaru explains that for a clear organ, dissolving lipids alone isn't enough. Yeah. Without lipids, the tissue would just collapse, so we need to maintain the structure of the tissue. I was gonna say, okay, from the sounds of it, this doesn't sound like something you can do while it's alive. This definitely sounds like this would be, like, insane to do while any kind of creature is alive. Alright, let's see. So, and it's true, like, your lipids are very important for structures and everything, so you can't just dump it. So, they did this by infusing acrylamide, a chemical that links proteins or molecules in tissue together, forming a gel. I'm trying to think if I remember how acrylamide works. I'm pretty sure that's also standard. But if it actually replaces the lipids where they're at, or if it's kind of like just gluing everything together. This means that they can dissolve the lipids and leave the structure intact. Gradinaru's group called this technique PACT for passive clarity. It does take a while. Dissolving out the lipids in the whole mouse or rat takes 1 to 2 weeks. Damn. Alright, that's a process then. Oh god, imagine if something went wrong during that. Because I assume they can't just dunk it in a solution and leave it. I assume like every day or every few hours they have to, you know, maybe change the solution or something. The choice of detergent is crucial in this process. Many other clearing techniques produce beautifully transparent organs, but the detergents often dissolve other chemicals as well, including fluorescent markers. Oh, okay. Fluorescent tags can be used to label cells or specific proteins for an experiment to compare different conditions and observe change. So like I was talking about earlier, that's how we can get all the beautiful colors underneath a microscope, is with those fluorescent dyes. Scientists need those labels to remain nice and bright in a cleared specimen. Previous techniques struggled with trying to find a clearing detergent that would efficiently render all the organs of the body transparent while leaving the fluorescent tags alone. Clarity was one of the best, leaving tags intact while clearing out a whole brain. Yeah, I'm trying to think of that. So, because it's like, you know, I guess you can try to wash out all the detergent afterwards, but there probably would still be leftovers that would mess with all your fluorescent tags and dyes. Let's see, this 3D image is a mouse kidney that's been rendered transparent with a new technique. Interesting. There's also the sheer amount of time these processes take. A single organ is small and detergent can be diffused through relatively quickly, but a whole body, even a small mouse body, could take far too long. Naru and her group realized that the best option is perfusion. This involves pushing a chemical, such as your detergent, through the circulatory system of the animal under continuous pressure. God, tell me you don't shove detergent into this mouse while it's alive. Tell me these mice are dead already. Circulatory system runs all through the body, so the detergent gets everywhere. Scientists call this method PARS, for perfusion-assisted agent release. I think I opened up the paper here, or was this for... This might have been for the other article, I'm not sure. Let's see, what's your methods? Buffers, permeability in your brain. Models, whole body staining. Okay, mainly based on the setup for pumping the pre-treatment solutions and immunosing buffers through the mouse heart and vasculature to perfuse the whole body. So I talked about using the... Okay, they keep referring it to his body, so I don't think they actually, like, shove a tube into this mouse's heart and they're like, alright bud, you're about to have the worst last, hours of your life. Okay, let's see. At the mice. Where we go, 3-month-old. Absence of bacteria is confirmed in germ-free mice. Mice have been used for further experiments. I just want to see and like confirm. Is there a point where they sacrifice? That's the technical term that we use instead of murder or kill. Is there a point where they sacrifice the mice before doing that? Okay, deeply anesthetize. Oh no. After washing the blood out, use the mouse bodies or skin transferred to PBS after post-fixation. Well, let's not think about it too hard. Okay, so let's see. Alright, so once the tissue's been made transparent, Gradinaru's lab embeds it into a solution that makes it easily viewed and photographed. The end result is a see-through mouse or rat that can be labeled with fluorescent dyes. Tissue is so clear that the researchers have been able to detect single strands of RNA, Gradinaru says. Interesting. They have also used the technique to isolate and label cancer cells from a human skin cancer biopsy. Gradinaru has high hopes for the method. We can obtain better maps for a neuronal network, she says. A transparent whole organism is particularly useful to map the distribution and identity of peripheral nerves at their target organs. Yeah, this sounds good as like a, definitely as a post-mortem type of technique. But I was hoping, I mean, you know, there are other methods that we can do for it, I suppose. I was hoping there would be something like, say, you know, someone gets brain damage or something, and it's like, okay, well, what nerves have been affected in the system? I was hoping it was going to be something like that, where we would be able to detect that and run through the entire nervous system to see that. That's alright, I guess. Let's see. Mapping the final endpoints and nerves in detail could produce better methods for electrical stimulation, which could aid in therapies for disorders such as migraine, epilepsy, and depression. Deseroth also hopes that the technique can aid in studying whole body progression of cancers and give scientists better understanding of how mammals develop and age. Yeah, it'll be handy on animal studies for sure. While clear mice and rats can provide amazing detail, they also have limits. The main limitation: the technique requires a dead mammal. Okay, so it does have to be dead. Cool. We're not torturing the hell out of this thing. Because all the tissue is post-mortem, we cannot investigate phenomena as they happen in real time. We can just get snapshots. Granari explains. Being able to clear out a living animal would allow scientists to see whole bodies, functioning all the way down to the molecular level in real time. A fully transparent live mouse or rat, and its accompanying horror movies. Okay. Is probably a long way in the future. Even so, the current technique allows scientists to better view the body than ever before. A bizarre, gummy-looking mouse may look strange, but represents more new and impressive opportunities to enhance our understanding of our bodies and ourselves. Yeah. Okay. But pretty good technique. What was this? 2014 again? Yeah. Interesting. Just look at that thing. I wonder how it feels too. I wonder if it feels as squishy as it looks. What was it? There was something else I saw. Cause yeah, I'm trying to think if I mean, you know, so there are similar like sort of studies that we do on things like this. Take for example, zebrafish. We actually use zebrafish for certain, or actually for quite a few like neuronal studies and stuff, because you can look inside, you can look through their bodies, so like you can add your dyes to them, and then you can actually see like the development or the location of all the nerves in their system. The zebrafish are pretty useful with things like that. And I'm sure there's probably some other like transparent or semi-transparent animals that we also use. Look at this guy. Okay, so it's more the larvae, I guess, that are transparent. But alright. So, yeah, see-through mice are nothing new. But existing techniques to image their insides can be expensive, time-consuming, or don't hit the right target. Now, study shows that chemically removing cholesterol, an essential component of cell membranes from dead mice, creates sponge-like holes in tissue without destroying them. That means tailored antibodies can move through the holes to infiltrate every corner of the body and bind to proteins of interest to make entire anatomical features visible under fluorescent light. So basically just makes it easier to be able to add the dyes to see its tissue. The technique, dubbed a wild disco, man, loving these names that these people are given the techniques, gives scientists an extraordinarily thorough peek under mouse skin to create body-wide anatomical atlases. It's a bit like Google Maps, says Ali Irtuk, sort of, a neuroscientist at Helmholtz, Munich. But in places of cars driving around to record every street, antibodies act as street lamps to illuminate scientific landmarks. Such maps could help researchers train artificial intelligence programs to simulate bodily processes in mice. That could be good, honestly. The algorithms would aim, for example, to simulate how a drug travels through blood vessels or predict the path of genetically engineered immune cells for cancer therapy. Computational simulations of biology may help researchers move away from doing animal experiments, he says. No, see, this is... I feel like it's got such a stain in my mind now, but this is actually something that artificial intelligence would be amazing for. Or just like machine learning, to teach it like, hey, this is how a mouse or like whatever's nervous system works. And then we can come up with different simulations of, you know, like whatever study or technique or something. Although, depending on what it is, say like drug therapy, I'm sure we can't just simulate that. That would probably still require some pretty solid evidence. And I don't think AI would immediately, like, take away animal experiments. But, you know, it'd be good to not have to use as many mice for said experiments. But again, I feel like we would need a more solid, like, do the simulation, then, like, prove it with the mice, is what I would imagine. Alright, so the wild disco imaging technique turns a mouse lymphatic system into a kaleidoscope of blues and yellows. Blue indicates the lymphatic vessels and organs are closest to the camera, while yellow marks ones are deeper inside the body. Okay, so it's not... Okay, phrasing. Oh, welcome back. There weren't any, like, huge pictures I showed. I just showed, like, some transparent zebrafish. But yeah, like, phrasing... So not closest to the camera. It's not the camera that's made it closer, but it's just the fact that yellow's deeper inside the body. So, like, that's why, say, like, you know, because it would make sense that, like, if it was in proximity to the camera, like, this would all be blue, this back here would be yellow. But that's saying, like, the blue here is, like, kind of closer to the outside, yellow's on the inside, and the green sort of, I guess, in the middle. To make the mice glow, your two colleagues pumped cholesterol-removing chemicals along with antibodies tagged fluorescent molecular markers into the dead animals via the heart and blood vessels. Proven tactic that allows liquids to traverse the entire body. You know, I wouldn't be super surprised if they did use them live though, because just the... I think the only maybe downside, when you use the heart and you pump things in yourself, is it could damage some of the blood vessels, and depending on what you're trying to do, maybe you want to avoid that. But, you know, probably better to do that rather than shove detergent into a living mouse. Alright, so then the team made all the tissues transparent, and mice were ready for their close-ups. Man, I love technology for stuff like this. It's just like coming up with these techniques of how to develop a method or a way to visualize a nervous system. You know, it's just like this is some good, like, innovative thinking. Let's see. So, one of the resulting maps depicts a comprehensive view of the network of nerves that run beneath the skin and around organs. Another shows off the lymphatic system, an open network of organs and lymphatic vessels that helps fight off pathogens and transport cellular waste for removal. These blueprints yielded some scientific nuggets. Images taken of the nervous system, for instance, showed that mice with no microbes in their body had underdeveloped nerve networks in their guts compared with normal mice. Okay, so I think what's talking about, in the technique to do this, they have to be germ-free, I suppose. Which there are, quote-unquote nude mice that hold on. I gotta be careful and, you know, I gotta think about what I'm searching. Make sure nothing weird or wild comes up. Okay, cool. We're good. Okay. So here's nude mice. Yeah, usually you don't have, like, any hair. What did I just hit? And and immunodeficient, so they don't have any immune system. And they're kept under, like, extremely strict, what's the word I'm looking for? Their environment is just kept extremely clean, obviously, since they have no nervous systems of their own. And that's usually so you can, like, inject, like, tumor cells or something to or for whatever your research is. So I think these have to be new mice, but I can double check. That might be. I'm pretty sure they said they just use normal mice, but I'm not sure. So let's see. But that is interesting that without the gut microbiome or without other microbes like around in the body, it will actually underdevelop nerve networks. Because again, you think about it, it's like everything has evolved with bacteria. So if we get rid of bacteria, it would actually be more detrimental to us than helpful. You know, bacteria play an extremely important role in all of our lives and in all of our bodily processes. Let's see. In mice with cancer, the team documented clumps of immune cells inside and near the tumors. Wait. Tumors. Oh, okay, so they did. I missed that. They did put tumor cells in these mice. Okay. Okay, so clumps of immune cells inside and near the tumors. The role that these clusters play in controlling cancer is unclear, Zurich said, but Wild Disco offers scientists ways to investigate. Wild Disco might also help with studies that focus on animals other than mice, says Yujun Su, an imaging specialist at Howard Hughes Medical Institute's Genelia Research Campus in Ashburn, Virginia, who is not involved in the work. Scientists working with fruit flies, frogs, or zebrafish might use a similar recipe, he says, to take clear snapshots of their anatomy. Yeah, fruit flies you can see through, I think, relatively well as well. I'm curious about the frogs, though. So let's see. Immune cells, pink, cluster inside and near a mouse's cancer tumors, olive. And see, they kind of... another fun term that we use a lot, co-localize. The pink and the olive will overlap each other. How the cells might affect cancer progression is unclear, but Wild Disco could help researchers investigate. Okay, so that's it. It's just showing the relative technique. You know, I'm willing to bet as well that like for some like microscopy pictures, you don't actually care as much about like what you'll see from it. You're just like, let's pump the shit, like, or let's like really brighten up the colors. Like, it doesn't matter if we use this specific imaging for this specific image for anything. It's like we just need to get a pretty picture for, you know, like papers and stuff to be like, ooh, look at this. Like aren't you attracted by all these pretty colors? Isn't that neat? But cool. That'll be, yeah, in another few or 5 years. Be cool to see what else we've been able to uncover or discover with this. So I'm curious. I wonder if there's any animals that this technique would not work on. Or I wonder if any people would give their, I guess, like, consent to have this done to their body. Can you imagine, like, what, it said, like, a week that these mice had to sit in the solution? So can you imagine having to have a huge tub that a human body would probably have to sit in for, like, a week or a couple of weeks or maybe even a month? And by that point, I don't know if it would actually, like, be beneficial or if it would just be worse. Yeah, that was pretty good, pretty interesting. All right, next, 2 of 4 that I planned. You know, I'm thinking, with these science streams, I want to, we can really talk about anything, you know? I just pull some random stories, but if that ever brings us to a tangent where we want to go off on, like, some other thing we heard about recently or something, like, we can do that. We can just pull that up. That way it doesn't feel so rigid, you know? I hate very formal presentations and things of, like, we're only going to talk about this. Like, we can talk about anything. But yeah, this person, Elise G's brain is fabulous. It's also missing a big chunk. So this is the MRI scan, I think, of this person. And she's literally just missing this entire chunk of her brain on the left side. And, you know, we'll get into it, but a brief overview. Like, it said she was completely normal. Like, you would never be able to tell that, this is the case. And I was like, okay, cool. I'm always down for, like, wild brain development. How it's just like, oh yeah, like, you're missing this. See, isn't the human body fucked? Like, some people, they'll get, like, a little nick or something on their brain and, like, bleed or die or whatever. And some people, it's just like, oh, like, you don't have this entire chunk of your brain and you're fine. You're perfectly okay. Crackhead? No. Not in this case. I believe this was actually, or theoretically, was when she was, born, actually. Or, like, soon after. So it's just so weird. And, you know, there's the, what is it? There's that classic case of the guy that got the railroad spike or the pole or whatever to go through his head. And, like, he lived fine. It just changed his entire personality. But it's just, you know, it's one of those things like, man, what the fuck? You know, it's almost maddening of, like, I can't believe this person's doing so well. Of course, I don't know if this will have complications in the future for her, but we'll see. We'll see what they say. The most interesting brain project participants featured stories in shortened forms and their names and their pseudonyms to protect their privacy. Fair. You never guessed that Elise G has a black hole in her brain. Meet her on the street and be impossible to tell that she's lacking a chunk of neural tissue about the size of a small fist. Looking at her brain scans is a different story. It's as if someone has knocked over a bottle of ink. The darkness pools inside her skull near her left ear, a puddle of fuliginous black. Inside the splotch, there's no white matter or gray matter, no blood vessels or tissue at all. Elise says, you don't have to be a neurosurgeon to spot what's different about her brain. There's a big honkin' piece missing. love the usage of the term honkin'. Yeah, so here's the sacked MRI slices and just, yeah, there it is. Just a solid left part missing. Scientists can't say exactly how it happened. It's possible that sometime long ago, perhaps due to stroke, before or shortly after birth, a portion of Elise's brain died and then ultimately disappeared. So, like, during development, it's possible there could have just been some weird blockage or something. And as a result, obviously, those nerves don't develop, but everything else develops around it. What I am surprised about, and it does say here, leaving behind only liquid, brain tissue sought for a fluid-filled void. Her sister has one too. I am surprised that, I guess, just liquid sits there, rather than the nerves developing in that section and taking up that space. But Lisa and her sister, Martha M., who are not using their full names to maintain their anonymity, look and act perfectly ordinary. Each lacks most of a temporal lobe, and each in a different hemisphere. Oh, interesting. Okay, I'm not as good with my brain thing, so I didn't remind myself what temporal lobe does. Managing emotions, processing information, storing and retrieving memories, and understanding language. Okay, so I am curious if there's any, like... I mean, I guess you have your right temporal lobe, so it's maybe okay. Let's see, Elise is also missing part of her brainstem. Damn. Okay. A woman or two of who knows how many people live in their lives without brain structures generally thought to be crucial. Martha, now age 59, damn, didn't know her brain was different until she was a teenager. Elise, who will turn out 61 this year, found out in graduate school. two sisters, two brains, two black holes. When MIT cognitive neuroscientist Evelina Fedorenko's team first learned about the duo, we were all kind of blown away, she says. Elise emailed Fedorenko her brain images in 2016. I'm curious why they needed their brain image, though, if it was a problem with that. Let's see, decades after the void was discovered, she had read an article about neuroscience research at MIT and was curious if scientists would be interested. I love that she's just like, oh, these people, like, read an article about neuroscience at MIT. It's just like, oh, you guys work with brains, right? Do you want to do stuff with this? She said, I'm missing my left temporal lobe. Do you want to study me? That's so good. I mean, to be fair, as far as we're aware, she is a bit of an anomaly, so, you know, if someone's needing a project or whatever, I feel like they would just be like, sure, fuck it. The left temporal lobe is generally thought to be essential for speech and language, when Fedorenko, who trained as a language researcher, was intrigued. Okay, yeah, it's like, okay, yeah, I'm curious because I'm like, language research, and again, this woman just read like, oh, you do brain research, right? But it's like, neuroscientists don't just do the whole brain, or like, you know, it's not an everything field. You kind of have to funnel it down a little bit what you want to study. But I guess being offered that, yeah, it was just like, her lab had studied people like Elise before, but I'm a very adventurous scientist, she says. Her team brought Elise to the lab for tests. Fedorenko didn't know it at the time, but those first studies would set in motion a whirlwind that would alter the course of her research. Which can happen. Her team's findings would ignite media attention, prompting even more people to send along their brain scans. What started as a single case study has now snowballed into the Interesting Brains Project. Yeah, okay, what? Oh, they actually have all the pictures. Wait. Oh my god? No way? The entire front half of someone's brain? Alright, I gotta learn about Interesting Brains Project real quick. A substantial fraction of the population lives with brains that look different from a typical brain. Affected by stroke, physical damage, cysts, buildup, fluid. Okay. Let's see, we believe that studying the functional organizations of atypical brains in both individuals who experience some cognitive difficulties and those who live symptom-free can reveal critical new insights about how our brains work in general, what they are capable of in the face of tissue loss or displacement, and maybe even how different brain components have evolved. Which, like, brains, honestly, to me, one of the biggest, like, black holes of knowledge. Absolutely. Brains are so confusing. Okay, we're interested in diverse questions, including the following. When does the language system emerge in / remove to the right hemisphere? Okay, so they still do language stuff, then. How does the brain cope with having less tissue to work with? Is language more likely to share resources with some function than others? Which, you know, yeah, this is a good question, but it always makes me think of this thing that's like, oh, like, you use 10% of your brain, or you, oh my god, she's unlocked all of her brain power, sort of thing. But that's, as we scientists know, that's bullshit. We don't use a portion of our brain, we use all of our brain. But it is curious how the brain can not concept, what is the word? Man, I always forget vocabulary. But essentially how the brain can function and make up for the loss of function of whatever brain part's missing. Let's see, language more likely to share resources, some function, others. Why are some brain atypicalities associated with cognitive difficulties while others are completely asymptomatic? I wonder if that has to do with the age that these brain parts would be damaged. Let's see, what determines whether or not loss/compression of brain tissue would be associated with cognitive symptoms? Alright, let's see what they found. Okay, so they put out a paper, they put 2 out this year. Alright, investigated brain mechanisms support reading ability. See, typical individuals have an area on the bottom of their left temporal lobe that is highly specialized for processing letters and words called the visual word form area. This area develops when you learn to read. Okay, so... Wait. Okay, so EG is a person. EG... Oh, this might be Elise, actually. Elise G. Yeah, so EG's language system resides in the right hemisphere. We have expected to find I don't actually know what this is. It's a VWFA. Oh, visual word form area. Okay, so that's just the region. So we expect to find a VWFA on the bottom of the right temporal lobe. Strikingly, EG does not have a canonical VWFA, not on the bottom of the right temporal lobe. Not in the small remain portion of her left temporal lobe, not anywhere else. So I wonder what determines that specific area visually. Instead of having a specialized area, EG appears to rely on distributed representation, such as small bits of the brain, voxels, that show strong responses during reading are interleaved with voxels that show preferences for other visual categories. Best of our knowledge, this is the first report of intact reading ability relying on a distributed neural code. So it sounds like she just straight up doesn't have this like specialized portion of the brain. So her brain instead relies on the part, instead of like developing something for, I guess, language, it sounds like. You know, I'm speaking from someone that does not have a brain neuroscience background. For, you know, caveat. But yeah, relies on the part of her brain that just looks at visual categories. So I guess she kind of can, like, in a way, categorize words and how they look or something to try to process them or to try to read, which is pretty interesting. Bioarchives, this is probably still in preprint and probably hasn't been put out yet. Investigate the organization of the auditory cortex and visual, also EG. Wait. Oh, these are all with the EG. Okay. Who's missing most of her left temporal lobe, her sister, MM. Yep, it's definitely Elise G. And their neurotypical sister, TG. Okay, so I guess TG has a full brain. No missing areas. Let's see, these results suggest that development of the auditory cortex in each hemisphere does not depend on the existence of the other hemisphere, highlighting the redundancy and equal potentiality of the bilateral auditory system. Okay, so they may be missing like one side, but the other side still has the auditory cortex. Okay. Well, cool. Very neat. If you think you have an interesting brain, please get in touch. I have no indication that my brain would be interesting, to be honest, so this would be cool. Let's see, take part in a 2-hour fMRI scanning session. During the session, you would lie on your back. So complete some behavioral tests outside the scanner, 4 hours across 2 days. Okay. I do wonder how many child participants they've had. Let's see, including the following conditions: childhood strokes, cysts, hydrocephalus, some cases of resuscitation, or recession. Anyways, let's see, what else do we have? So, yeah, they started the Interesting Brains Project. By the end of this fall, the project will likely have scanned more than 40 people with atypical brains. In many cases, participants are missing entire brain regions, and like Elise, they didn't find out until they were adults. That may be a tribute to the brain's flexibility, its ability to change and adapt, including its redundancies, Fedorenko says. Like backup generators, some brain areas can kick into gear if others get injured. Close look at cases like Elise's could help scientists better understand how our brains cope with damage and why some kinds are worse than others. Man, see, isn't the brain fascinating where it's just like, at least when we're still developing, something gets fucked up and then, compensate. I think that was the word I was thinking of earlier. How the brain can compensate for another part missing. So let's see, for now, Fedorenko's team is focusing on language aspects of high-level cognition, such as a person's capacity for general reasoning. But the effort could also offer insights into the organs of the brain more broadly, might one day give doctors a better sense of what outcomes a person with a brain injury might expect. That's what Elise and Fedorenko hope anyway. Before she started working with the MIT team, Elise says, "I felt like my brain was something that needed to be cured rather than celebrated. Not anymore. These days there's a different word that comes to Elise's mind when she thinks about her brain. It's fabulous," she says. Hell yeah. Again, I do wonder about the, I'm curious if the, like, obviously it seems like her brain is fine with it missing, but I'm curious about the fluid that can, I guess, build up or whatever there. Technically, I mean, she could, like, get a tumor or something, and if it's in that area, would not, like, pressurize her brain, hopefully. Not push up against her skull. So MRI turns up atypical brains. Elise first learned about her atypical brain after an MRI scan in the fall of 1987. She was 25 years. I wonder how many people could have brains like this, and then like had an MRI scan or anything, and they were just like, oh, this must be a mistake. Like this can't, like you can't possibly be missing an entire section of your brain. So let's see, she was 25 years old in her first year of grad school in Washington, D.C., an avid reader and a whiz with a needle and thread. At an appointment with a neurologist at the George Washington University Hospital, Elise, who had been previously diagnosed with epilepsy despite never having a seizure. Interesting. Is that? Hold on. Epilepsy is... What? Why was she diagnosed with that then? Ain't paid her much notice, she says. Excessive abnormal brain cell activity. I'm just curious, like, does epilepsy mean you, like, are definitely gonna have seizures, or... Let's see. It seems like it's one seizures. I don't know. No clue why you choose diagnosed with it. Alright, oh, this might be after. I wonder if this is after the MRI scan. I thought this was just like in general. Let's see. That feeling evaporated when Elise came back for her results. Now she had the doctor's full attention. Okay. So they were like, whatever, you're fine. And then they're like, oh, wait, hold on. Holy shit. They lean forward in their chairs, elbows on knees, chins in hands, eyes laser focused on Elise. How do you feel? She remembers them asking. She felt like a lab specimen, like a frog they were zapping with electrodes, she says. Yeah, it is very interesting whenever... So a small tangent story that I have is my brother, he got diabetes, or he was diagnosed with diabetes, type 1, like 5? No, like, God, at this point, like 6 or 7 years ago. And my father was type 2 diabetes. Like, we basically already knew, like, what diabetes was, what it takes to sort of handle it. And we're just very, like, it's very funny. I've had it pointed out to me a lot, of how I'm not a very reactionary person. And it's just like, my entire family's like that. We just take things, and, like, with a straight face. And we're like, okay, like, sure. So, the doctor, like, was getting ready for when he was brought to the hospital, ready to have everyone like crying and weeping and everything. When he said like, "Oh, you've got diabetes," and then apparently because it was my brother and then my mom and dad. I was out of town at the time, and they were just like, "Okay." He's like, and then the doctor just kind of stared at him for a second, and then like was like, "Oh, okay," and then like moved on, because my parents were basically just like, what do we gotta do, like, just like, all right, he has diabetes, nothing we can do about that, so where do we move forward from here? So it's very interesting whenever doctors are just like trying to gauge a situation when it's like nothing much has changed, Anyways, what the doctors had seen in her brain scan, of course, was that blatant black hole. When detected in babies, it's the kind of lesion that makes parents fear the worst. In adults, strokes in the left hemisphere conceal people's ability to read and write and jumble their speech. The meaning of words can slip suddenly from the mind as if an eraser was scrubbed away a person's mental dictionary. Oh wait, meaning of words. I was like, oh no, words slipping from someone's mind, like me. Elisa's doctor, she remembers, was surprised she had more than a 5th grade vocabulary. Okay. She's in grad school at this point, damn. At the time, Elise felt sick and scared. She didn't know if the lesion was growing, if it foreshadowed early Alzheimer's disease, or if it was going to explode in my head. God, yeah, that's scary. A follow-up scan 6 months later eased her fears. The scan looked the same as the first. Her brain's black hole wasn't expanding. Elise never returned to his doctors, but she did get a second opinion that summer from her sister's neurosurgeon. He had operated on Martha when she was 17. Oh, okay, after she had noticed vision problems. The bulk of her right temporal lobe was gone, possibly due to a stroke in the womb. Fluid buildup in the brain was pressing against the nerves of her eyes, hindering her sight. Oh, they drained it and I went on my merry way, Martha says. She hasn't had the area drained since. Huh, I wonder if... Okay, so it got drained. I wonder if it needed to be drained again. See, this is what I wanted to know. I was like, I feel like there has to be some... like, why did you just randomly get an MRI and all that? So let's see, Martha's doctor looked at Lisa's scan, told her that as MRIs were becoming more common, doctors were finding other people with brains that diverged from the norm. He said, "We're seeing more and more deviations, and you've got one," she remembers. Let's see, Martha M., Elisa G's younger sister, is also missing most of her temporal lobe, but on the right side of the brain, a near mirror image of Elisa's lesion. Martha may have had a stroke while she was in the womb, later fluid buildup in the brain pressed against the nerves of her eyes, hindering her sight. Wait. Oh, okay. So Martha's the one that had the, sorry, I glossed over that. Martha's the one that had the eye problems. And Elise, yeah, sister's neurosurgeon. Duh. And Elise, I guess, hasn't had any, like, noticeable problems, maybe? Like all the interesting Brains Project participants tested so far, Martha scores at or above average on language and cognitive tasks. Interesting Brains Project is born. When Elisa and Fedorenko first met, Fedorenko was interested in how language areas wire up when a chunk of crucial tissue is missing. The plan was to peek inside Elisa's head using functional MRI, a technique that tracks blood flow in the brain. Functional MRI lets scientists see which parts of the brain are active while a person performs a specific task. For Elise, that means lying statue still inside the giant tube of an MRI machine while equipment whirs around her. Depending on the task, Elise may look at or listen to words, sentences, and stories, or see math problems or spatial puzzles. Occasionally, she'll press a button so the team knows she's staying alert. Outside the tube, the researchers have also tested Elisa's vocabulary, reading and writing skills, and intelligence. She scored near the top of every language test she took. I could have probably taken over the world if I had my entire brain, Elise jokes. Elisa's first scan with Fedorenko's team revealed language activity in the right side of the brain. The team reported in 2022 in Neuropsychology. Shifting functions to the right is one trick our brain uses to deal with damage on the left, something other scientists have reported previously, Fedorenko says. The team wondered if Elise's left frontal lobe might chip in too. Yes, Elise likes her left temporal lobe, Federico says, but her left frontal lobe, where language also typically resides, is perfectly intact. Is there any language going on there? Federico asks. But that lobe showed no language responsive areas at all. So I assume they do this via... I forget exactly what scan it is, but you know, you can literally like have someone do an activity or something and then look at what parts of the brain are lighting up. So I'll assume like they didn't see anything lighting up at that area. The findings hint at a neural order of operations for language development. Without Elise's left temporal lobe, language areas in her left frontal lobe couldn't wire up. Federico's team also revealed that Elise completely lacks a typical region for reading words. The team thought such a region might show up in Elise's right temporal lobe, instead she appears to tap into a network of neurons across the visual cortex. Okay, so like we read in the other thing, yeah, it just completely gives that process to the visual cortex instead. It turns out you can have perfectly functional reading visual machinery in your brain that's implemented in a different way, Federico says. Elise may be the first reported example of this. The findings from Elise's brain caught the attention of a reporter at Wired who wrote an article last year with an eyeball-grabbing headline. She was missing a chunk of her brain. It didn't matter. Then the emails started pouring in. The morning after the Wired story published, Federico's inbox was filled with cool brain pictures, brains missing all sorts of big parts, she says. In many cases, people found out about their atypical brains accidentally. Federico heard stories about people with neck extension going in for an MRI and finding out they're missing most of their right frontal lobe. Others, like Helena Santoro, have known about their brain lesions since they were little. Santoro. Santoro, a science journalist who reached out to Federico after reading the Wired story, had a stroke before birth and was missing her left temporal lobe like Elise. Doctors said Santoro would never speak and would need to be institutionalized, but month after month I surprised the experts, meeting all of the typical milestones of children my age, she wrote last September in an article about her experience for the New York Times. What I'm getting from most of this is, if you have a stroke when you're a baby, you're fine. Babies bounce, you know, as they say. Which is actually extremely true. I forget the name of it, but basically, babies are very, what is the right word? You could, like, cut... What I'm trying to say is, like, babies are still developing in things, so they still have all those developing tissues. So if you, like, say, cut the tip of their finger off or something, like, it could very easily heal from that. I don't know if their entire finger would grow back necessarily, but... you know, it would heal like perfectly fine and they'd be okay. And that's actually how we take care of certain things that can happen during development. So let me see if I can get a picture so if I can describe this better, because I do forget what this is called. Esophagus and trachea. Okay, so I forget exactly what this is called, but basically, let's see. Okay, so like right here, you know, you're, you know, obviously your throat can lead down to your esophagus or down your trachea, and there's like a little thing here to make sure that food doesn't go in here, just air goes in to your lungs and everything. But with babies, when similar tissues will touch, they will fuse together. So what can happen is, before the esophagus and trachea fully develop, these tissues could touch, like down here, and then that could create a, that could essentially create a hole right here, because the body will just think like, like you want to form here or whatever. And you know, obviously that's a problem to have a hole right below where the entrance to your esophagus and trachea are. So literally all they do is, for surgery, they just cut the two tubes apart, and then they'll stitch them up, and then within like, you know, X amount of time, a few months or whatever, the baby's like esophagus and trachea will just close back up the way they're supposed to, and then they're perfectly fine. Like, you know, remove stitches, whatever, and then they're perfectly normal. But I forget what the term for that is called. I guess this gives a better idea. But yeah, if a hole forms here, they can literally just cut that, and then they're fine. Like, it's fucking crazy the amount of things that can happen to babies, and they'll develop perfectly all right. But yeah, like, you know, have a brain issue as a baby, and then it'll be okay. It'll do fine. Have a brain issue as an adult, less fine. Less ways to heal from that. So let's see, where do we leave off? Okay, still not unclear why some brain injuries slide by unnoticed while others demand attention, says neuroimaging scientist Helen Carlson. Her team at the University of Calgary in Canada have worked with kids who have had early strokes in the motor cortex, the brain region responsible for movement. Some kids with large brain injuries have only minor weakness on one side of their body. Others with just a little whisper of a smudge on their MRI have quite profound disabilities for their entire life. Yeah, wild. That mismatch can be true of other difficulties too, including problems with language and general reasoning. It's one of several mysteries the Interesting Brain Project is pursuing. So, how the brain adapts. As of May 30th, the Interesting Brains Project had scanned the brains of 30 people. Some have holes in their frontal or temporal lobes, others are missing parts of their cerebellum, a brain structure involved in balance and movement. Still, other participants have brain matter that's squished up against the side of their skull. Scans show void that appear to have ballooned from the brain center. These atypical arrangements can stem from cysts, surgery, strokes, or excess fluid buildup in the brain. Some can result in a brain with much less neural tissue than normal, and sometimes the change can be abrupt. What happens when the brain needs to perform its same jobs but in a much smaller face, Fedorenko asks? What are the solutions our brains come up with when suddenly there's a lot less turf to work with? A community of scientists have already dug up some answers by studying kids who have had perinatal strokes. In some of these cases, during a baby's birth, or the weeks before or after, blood flow in the brain can cut off altogether, starving tissues of oxygen. Which, you know, I gotta say, fucking sucks that there are cases where you'll have a perinatal stroke, you know? Like, you haven't even actually consumed real food and you've already got a stroke. Which I assume, I don't know if that would make you more likely to have a stroke later on or not. The brain can adapt to this injury, but it's not a lump of clay with infinite potential. Not infinite potential, but yeah, babies are like clay. You can just shape those little bastards, and they'll be fine. Everybody thinks, oh, the brain is endlessly plastic, says Elisa Newport, a cognitive neuroscientist at Georgetown University in Washington, D.C. Hey, I actually went here recently when I went to that convention. I walked through here. But it tends to deal with damage in set ways. Newport worked recently with a group of 15 kids and young adults who all had perinatal strokes that resulted in left hemisphere damage in an area that processes words and sentences. In nearly every case, the participants' brains shifted language over to the same spots in the right hemisphere. It's as if the language region is flip-flopped from left to right, exactly the mirror image of what ordinary typical brains look like, she says. This pattern suggests that certain brain areas can serve as pinch hitters for language function. Let's see, ES's atypical brain may stem from, different. Okay, this one I'm curious about. Scientists think that GC may have developed cysts in their left hemisphere early in life. That could have compressed their left frontal, temporal, and parietal lobes. Though GC's language areas are squished, the cyst does not seem to have affected her daily life. GC is a speech-language pathologist. Wow. Cool. Let's see. ES's atypical brain may stem from an assortment of issues, including arachnoid cysts, a fluid-filled sac that forms between the brain and one of its membranes. Hydrocephalus, when excess fluid collects in the middle parts of the brain, may also have compressed ES's brain tissues. On top of that, ES is missing most of the tissue that connects the left and right hemispheres, a condition called agenesis of the corpus callosum. In her daily life, ES is a professor and researcher in the field of speech-language pathology. Wait. Okay, I was about to be like, is everyone here working speech-language pathology? Writer, speech-language pathologist. Okay, so two that are speech-language pathologists, and then this person's a writer. But yeah, pretty wild what you can live without. Hello, Rango. Welcome. We're talking about atypical brains, and how if you have some sort of damage that happens to you as a baby, causing you to have a large part of your brain not develop, you're probably going to be okay. But there's still a universe of more questions, Fedorenko says. She's curious if brain functions can overlap, sharing the same cortical machinery in atypical brain, when they might otherwise have set up shop in different locations. And a damaged left hemisphere doesn't always mean language moves to the right. Sometimes language function stays behind, surviving on the fringes of the damaged region, Fedorenko says. Nobody knows why that happens. Man, the brain is such a black hole of information. Carlson and colleagues reported this kind of adaptation in 2020 in pediatric neurology. The team was studying young stroke patients who had perinatal strokes that hit the motor cortex. Wait, did I already read this? Or perinatal something or other. Whatever. Okay. So Carlson says the Interesting Brains project is valuable because it can tell scientists more about plasticity in the brain at the individual level, how a specific person's brain has adapted to injury. Not every person's brain is able to bounce back. What scientists learned from the project, along with individualized neuroimaging, could help with prognosis and potentially rehabilitation. Perhaps if we can tailor intervention options to an individual brain, they might be more effective, Carlson says. Getting results will take time. Fedorenko's team is currently judging experimental logistics, including scanning a new participant every 1 to 2 weeks, performing behavioral tests and analyzing data. Still, they're seeing some interesting results, Fedorenko says, and hope to submit a paper this summer. She hopes the project can showcase the range of solutions our brains can, in some cases, employ to deal with a slow or sudden loss of neural real estate. Maybe, she says, the project's findings will help more people understand how different you can be and still grow up and do amazing things. What is a normal brain anyway? In an opening note for Rinko's 2022 paper in Neuropsychology, Elise wrote about her brain structure doesn't define her. You were just forced to play Fortnite? Unfortunate. Please do not call my brain abnormal. That creeps me out, she wrote. My brain is atypical. Sure. If not for her accidentally finding these differences, no one would pick me out of a crowd as likely to have these or any other differences that make me unique. Elise hopes the message comes through for doctors and research scientists. I want them to understand this is a person they're reading a paper about, not a disembodied brain in a jar, says. You know what? True. I was talking recently about how, again, some researchers really don't see people as people. They will just see them as like, you're just a test subject, sort of thing. Yeah, never played Fortnite, and I want to hold off on that as long as I can, because I think the longer I go without playing it, the funnier it is. One thing Elise likes about working with Fedorenko's team is that the research feels collaborative. Scientists rely on close partnerships like this to understand how the brain works under typical situations and how it may recover from injury, says Leslie Fellows, a neurologist at McGill University in Montreal, who studies how brain damage affects decision making. People with atypical brains can give us all kinds of great ideas we might not have thought about, she says. They have a unique vantage point. Elise, for example, experiences small hallucinations. Okay, so he does have a little something. She picks up whiffs of electrical fires whenever she's under a lot of stress. When I was in grad school, I could smell electrical fires 3 times a week. Damn, she says. Elise hasn't yet explored this brain quirk with Fedorenko and her colleagues, but she's open to their ideas for future investigations. I guess those are things that are in Fortnite then. Let's see. For the team's most recent study, reporting to preprint this year, Elise, Martha, and another sister, one with an ordinary brain, participated in hearing tests inside the MRI tube. Fedorenko's team went to find out how the left or right auditory cortex works when the other side is missing. You might think the remaining auditory cortex would have to be enhanced somehow to pull double duty, perhaps taking up extra space, says Tamar Rajev, a cognitive neuroscientist from Fedorenko's lab. That's not what the team found. In both Elise's and Martha's brains, activity looks completely neurotypical, Rajev says. That suggests there's some redundancy to the brain's auditory system, and that the development of one auditory cortex does not depend on the existence of the other. This is actually true in many organs that we've got. When, say, like our kidneys, you know? Obviously both will filter out blood, but if we have to remove one or one is missing, like the other one can pick up the slack, you know? Maybe it has to work a little bit harder possibly, but we can still be perfectly fine with just one kidney. And interestingly, with how our lungs work, we can also be fine with just like essentially a chunk of, well, not fine, I would say, because like you would have a lot harder difficulty breathing, but you can still live with like a chunk of your lung. And it's wild that our bodies are just made to live. It's crazy. Wait, what? Play what instead? Oh, I see. Against bots and things. Yeah, I was wondering, it seems like there's a lot of bots in Fortnite, which is, I don't know, weird. Ah. The good old battle pass. Oh, I guess it just throws you in there with bots then? Interesting. Alright, let's see. Okay. So Elise is curious what other insights Fedorenko's team will glean from her brain and the brains of fellow Interesting Brain Project participants. My brain is especially unique and interesting, she wrote in the 2022 paper, and I am excited that it can help neuroscientists understand the plasticity of the human brain. I'm actually curious if she got to partake in writing part of the paper, because I think that would be kind of cool. Let's see. That is, I will assume, not the whole paper. It doesn't even, nah, it doesn't list her as an author. Okay. Alright, well, cool. So, if you have an atypical brain, you too can go to the Interesting Brains Project and contact them about studying your brain. Let's see, they say anything else here? Search the language lab, TED lab. Alright, well, sweet. Anyways, moving on. Yeah, this one. So, microbe stops mosquitoes from harboring malaria parasite. Strategy could provide additional weapon against spread of deadly disease. So yeah, apparently there's some microbe that's stopping the parasite that's, you know, that mosquitoes are carrying that cause malaria. Yeah, and it was it was pretty interesting. And I think she said, but like, didn't want to be referred to as like abnormal and liked atypical better. But a lot of people, like, 2 people missing with parts, like, were language speech pathologists. The main person that article was about, she at least was in grad school when she found out that she was missing a giant part of her brain. So it's just cool and wild how much she could be missing and, you know, be perfectly fine. And one of the speech pathologists, it was almost like half of their brain. It was like only a little bit of their left hemisphere seemed to be there, or at least from that slide on the MRI. Right, so let's see. Malaria. Malaria kills more than 500,000 people every year, most of them children under age 5. Yet strategies to control the disease are real challenges. Mosquitoes develop resistance to insecticides, and vaccines provide incomplete protection. Now researchers say they found a new weapon. A naturally occurring bacterium that, when fed to mosquitoes, halts the development of the malaria parasite in the insect's guts. And yeah, this is one of those like constant fighting evolution type things where it's like, alright, we made an insecticide, and it's like, well, mosquitoes developed a resistance to it, and it's like, okay, we got vaccines. It's like, well, the malaria or the vaccine doesn't provide great protection, so malaria got past that too. So it's so interesting that just something randomly comes up. But let's see. The findings offer a complement to existing malaria prevention methods, says Caroline Bareilles Murray, who leads the malaria and vector research lab at the U.S. National Institute of Health. The approach has great potential to be implemented in areas where malaria is endemic, adds Bareilles Murray, who is not involved with the work. Researchers have tried to use microbes to control mosquito-borne diseases before. The virus-fighting bacterium Wolbachia pipiensis has shown particular promise against dengue fever in recent clinical trials and is already used in some areas of the world. But most methods for blocking malaria-causing Plasmodium parasites, which are transmitted by different mosquito species from dengue, have relied on genetically modified bacteria. Wait. Okay, so that's something we have been doing, is genetically modified bacteria. That's a major obstacle to regulatory and public acceptance, Bareilles Murray notes, given the unknowns of releasing edited organisms into the wild. Yeah, everyone hates genetically modified things because they're like, I don't want it to genetically modify me, and it's like, well, that's not how DNA works, is it? The bacterium in the new study, published today in Science, inhibits the malaria parasite without any genetic tampering by humans. Which, again, damn if you have been working on these genetically modified bacteria and trying to fight malaria. And then just a random bacteria pops up and is like, hey, I'll take care of this. And it's like, oh, great. Will you? So let's see, Janeth Rodriguez, scientific lead in global health medicines R&D at GlaxoSmithKline, and all colleagues stumbled across the microbe at a GSK research center in Spain. Yeah, it's a good find, and at least it gives like a more solid, I guess, approach, hopefully. After noticing the mosquitoes they were using for malaria research were getting harder to infect with Plasmodium, it turned out there was an unexpected microbe, a bacterial strain called Delphtia surahatensis, TC1, inside these mosquitoes' guts, Rodriguez says. It's not clear how the bug got there, though the species has been seen before in the guts of some insects, as well as in water, soil, and rarely hospital-acquired infections. But when the team fed the strain to other mosquitoes, it seemed to block the parasite from infecting them. Yeah, hopefully. I'm curious, let's see, they said, was this in their lab or a research center? Someone across it at a research center. Okay, so they were just, so it was the mosquitoes that they had raised for malaria research. So again, I wonder if this is just something that randomly came up as a result of the mosquitoes being used as a vessel for malaria. It's been seen before. Malaria researcher at the Johns Hopkins Bloomberg School of Public Health, his lab discovered TC1 was disrupting the plasmodium's growth in mosquito gut, where the parasite develops before moving to insect salivary glands. Compared with mosquitoes lacking the bacterium, insects with TC1 had about 75% fewer plasmodium oocytes, egg-like structures the parasite forms in insects' guts. I'm actually curious, what's the malaria life cycle? So I actually don't know too much about the bacteria of malaria. So let's see. The mosquito takes a blood meal. I don't want. Let's see, malaria. I don't know what the parasite's life cycle is like, and if it just gets taken up by mosquitoes, or if they how they move from mosquito to mosquito. I don't think I'm gonna get the information I want here. But gross. Seeing how the malaria just pops our cells. Alright, anyways. So, mosquitoes lacking bacterium had fewer plasmodium oocytes. Let's see, disrupting plasmodium's growth in the mosquito gut where the parasite develops before moving to insect salivary glands. That's so weird that it moves from the gut to the salivary glands. So experiments in rodents revealed that disrupted plasmodium growth led to reduced transmission. Only 1/3 of mice bitten by bacterium-carrying mosquitoes became infected, compared with 100% of mice bitten by regular mosquitoes. You are so right. I'll take a drink now. The research identifies TC1 as an attractive tool for malaria prevention, Jacobs Lorena says. Mosquitoes only need to eat a little of the microbe to become colonized, and once inside the insect, the bacterium stays there, continually blocking parasite development, he explains. What's more, TC1 doesn't appear to affect the mosquito's survival or that of its offspring, meaning the insects are unlikely to evolve resistance to it. Hopefully. The Johns Hopkins researcher... I do wonder if it would be... I think there have been, as far as the genetically modified bacteria, I think there have been efforts to spread those among mosquito populations and stuff, so I wonder how hard it would be to spread this particular bacteria to mosquito populations. Let's see, hold down how the bacterium hurts plasmodium by secreting a molecule called harmane, a compound found... Okay, so we know why it stops it as well. Compound found in plants used for traditional medicine in some cultures. Feeding this compound on its own to the mosquito or just getting the insects to walk on it hobbled the parasite's development, though the effect wore off after a day or so. Okay, interesting. But I guess since this bacteria is constantly secreting it, maybe, then that's why it would keep up the effect. Findings suggest harmane might be used to treat bed nets or other surfaces mosquitoes come into contact with, Rodriguez says. In a last round of experiments, the scientists teamed up with researchers in Burkina Faso to test the approach on mosquitoes in a roughly 10 meter by 10 meter by 5 meter netted enclosure designed to simulate the real world with plants in breeding areas. I saw, let me see if I can find one. I saw a, malaria research lab recently, and I fucking hated it because they had, I don't know if I'll find one. They had, a little box, a little, netting box or something, and it was filled with mosquitoes. And what this lab was doing was they were trying to make the... I think it was... Oh, they're researching bug spray. That's what it was. So they're like, look, I put this bug spray on and then the mosquitoes won't bite me. And I watched this scientist, she, takes her hand and she just puts her like, or her whole arm in this netting enclosure with the mosquitoes, and I was like, I fucking hate that so much because it was filled with them, and they weren't biting her, so I was like, hey, it's working, but that makes me so uncomfortable. But like, I don't even know how to search for a picture of that, you know? Oh, you know what? This is kind of like what it looked like. It was like a little thing like this. Or sort of like this, and just imagine sticking your hand in that fucking thing. Oh god. No way. Look at this. I hate this so much. Anyways. Yeah, keep that in your nightmares. So let's see. Leaving out balls of cotton wool soaked in sugar and TC1 was enough to colonize some 3/4 of the mosquito population with the bacterium overnight. When these mosquitoes fed on blood from people with malaria, the bacterium blocked the parasite's development just like it had on the in the lab. It's a really impressive piece of work, says Grant Hughes, a vector biologist at the Liverpool School of Tropical Medicine, who is not involved in the research. He praises the study's use of multiple experimental approaches to understand the bacterium's effects on malaria transmission, and says the identification of harmine as the plasmodium attacking compound is particularly useful for future work. Barillis Murray highlights another important finding from the study. Unlike some gut bacteria, TC1 isn't passed between mosquitoes. Ah, okay. So that means we would actually have to... Yeah, that's actually even more annoying then. Can't just have mosquitoes pass it to each other. Although this could be a limitation down the line, it's a plus from a safety perspective and may make the path to regulatory approval easier. When you are ready to deploy massive amounts, you would like the bacterium to spread, she says, but before you do that, you really want one that doesn't spread too much. That way, she says, researchers can evaluate its efficacy and safety before it takes over a whole population. I guess that's fair. I guess that makes sense, doesn't it? To make sure that they spread in a safer way. Rodriguez, who along with another GSK researcher is named as co-inventor on a patent related to discovery, says an eventual product could take the form of bacterium-containing powder or even harmine itself mixed with sugary baits to attract the insects. For now, though, the researchers are running additional experiments to test the method's safety, making sure, for example, that the bacterium doesn't harm other insects such as bees. Hughes says he hopes the field will be spurred to look for other gut bacteria that might have similar effects. There are so many different bacteria and microbes that are associated with these mosquitoes, he says. The implications for using these microbes to control disease are very promising. Alright, cool. Well, yeah, I guess we'll see if that actually does help with malaria, and if it does, awesome. You know, the only issue would be we would have to spread the bacterium kind of manually. Alright, on to the last article for today. And this is something that I'm a little excited about. But scientists have recently released the Human Pangenome Reference. And I'm sure this article is going to talk about it. But basically, you know, we had our first human reference genome in 1990. I should know this. 8, maybe? Hold on. Let's see. Nope, not 2022. Was it 2003? Or was it just the... Am I thinking the announcement of it? Hold on. Now I'm going crazy. Was it 2000? I swear I thought it was like late 1990s. Okay, the project launched in 1990. And then... initial rough draft was in 2000, 2001. And final sequencing was in 2003. But... So... with our genomes, we have many different varieties, variations, and such. in our human genome. I'm starting to lose my voice. So, you know, you say like, oh, cool, we've got the mapping of the human genome. Now we know all the letters, all the sequence of the human genome. But you kind of ask yourself, like, whose human genome? Because, you know, your genome's not going to match this. You could have like a huge percentage, and I say huge relatively, like huge number of bases and such that differ from this. So with this, essentially, what this does, the human pangenome, is that shows multiple reference genomes that you can look at, and that will cover different diverse backgrounds. You know, I think it's only like 40 or so, but this is just the first release. But it's to sort of cover like, and you know, I don't know the demographics of the first draft of the human genome, but this is supposed to be much more encompassing and be much more applicable to everyone rather than just like a few, you know? So let's see, the first human genome sequence generated by the Human Genome Project 20 years ago. Oh yeah, damn, I guess it is 20 years. Hold on. I missed the anniversary. Shame. We could have done something fun for that. Has served as a reference for researchers to use in their genomic studies. The first human genome reference sequence did not represent a single person, rather it was assembled with genomic data from about 20 people to be a general-purpose tool, against which scientists could compare other human genome sequences. Yeah, so, you know, 20 people doesn't exactly cover the now 7 billion plus population. Over the years, researchers have regularly improved that reference sequence, fixing errors and adding missing pieces, because there has been, okay, I guess this is literally going to say it, but there's been better sequencing methods to be able to replace those missing pieces. So recently, new genome sequencing technologies, particularly long-read DNA sequencing methods, have made it possible to read longer stretches of DNA at once. These technologies allowed researchers from the NIH-funded Telomere to Telomere Consortium to fill in the missing gaps and create the first complete human genome sequence in March of last year. Damn, really? I didn't know it was that recently. While the original human genome reference sequence has been regularly improved, it still consisted of genomes from only around 20 people, which does not adequately represent the diversity of our species. On average, any two individuals' genomes are more than 99% identical, but that still leaves millions of differences. Yeah, so if you take your genome, which is about, what was it, 3 billion? Or is it 3.5 billion? I should know this. I've been out of it for too long. But billions of base pairs, 1%, yeah, that's gonna leave millions and millions of bases that are still different. A more inclusive human genome reference sequence will allow researchers and clinicians to identify and understand the small genomic differences that make each person unique, providing genomic insights that could be relevant for health and disease management. Last month, the NHGRI-funded Human Pangenome Reference Consortium released a high-quality collection of human genome reference sequences, which together comprise a human pangenome reference. Encompassing genome sequences from 47 people of diverse ancestries. I'm curious how diverse. The Human Pangenome reference captures significantly more population diversity than the previous reference sequence. Specifically, a set of individuals previously studied in the 1000 Genomes Project had their genome sequenced with very high accuracy. You know, I'm sure our sequencing technologies are just going to keep improving. So this is probably a thing that needs to be revisited like constantly, to be like, oh, we got some new technology, like let's make sure, you know, let's check our accuracy and let's check let's add new info. The current human pangenome reference consists of genome sequences from 47 people with different ancestral backgrounds, including people from Africa, Asia, the Americas, and Europe. Scientists in the consortium aim to increase the number of represented genomes to 350 by mid-2024, when the project is expected to be completed. The new human pangenome reference adds 119 million bases to the existing human genome reference sequence. And one reason why it's kind of difficult to fully reference the human genome, just one thing off the top of my head, is there are these sequences. God, I should know the term for this. There are parts in our genome that will essentially quote-unquote jump around. So it'll be like a small-ish, relatively number of bases, and they'll actually kind of move around in the genome just due to the, I guess, machinery in there and stuff. And so that can be kind of a pain in the ass when you're trying to be like, oh, like it's over here, or it's over here, depending on which genome you're looking at. There's also many parts in our genome that repeat just the same sequence over and over and over and over again, and machines don't really like that. It makes it kind of hard to figure out from the way that the bases are read what exactly the sequence is supposed to be if you're just, say, having like A and T repeated over and over and over. So with technologies now, there should be ways that we can kind of start to overcome that and make it easier to look into. So the main human pangenome reference paper is one of several papers published in Nature that report findings generated with the new human pangenome reference. This included developing a map of single nucleotide variants within segmental duplications and observing patterns of recombination between different parts of chromosomes. As a truly collaborative effort, the Human Pangenome Reference Consortium consists of researchers from various institutes around the world working to create a more complete and sophisticated collection of reference genome sequences. As the construction of a human pangenome reference is still ongoing, researchers from the consortium are working hard to add more genome sequences and improve its quality and diversity. Importantly, the Human Pangenome Reference Consortium has an embedded ethical, legal, and social implications research group, okay, that's pretty good, to guide informed consent, sample prioritization, regulatory policies, and collaborations with international indigenous communities. This group ensures that the collection and use of the genomic data are ethical and that communities around the world are engaged with the research process. So, yeah, no worries, they're not just gonna take your DNA and then use it without your consent. Generating the Human Pangenome Reference is part of NHGRI's ongoing effort to account for global diversity in genomics research, which is important for advancing and implementing genomic medicine in an equitable way. Over here, press release. But yeah. So that's honestly, like, it seems like a small thing, but that's pretty great for future sites. Let me see if I can find it actually. Because, like, you think maybe the human reference genome, like, maybe that would be not super huge important, I guess. But I want to see if we can find the, if I can find the original paper. So we'll start here to 2004. If I can find the original paper, I want to see how many citations it actually has, or at least according to Google, It's like this one, like, here we go. 19,352. Quite a bit. A lot of people. Yeah, this is like, let's see, this is 2001. Yeah, so this is one of like the original papers talking about everything that they did with referencing the human genome and everything. And I feel like even 19,000 seems like a small number of citations, but with everything aside that, and then everything that gets cited from those papers, like it's kind of a big tree, I feel like, of citations with the human reference or the human genome. But yeah, that's basically all I had today. So that's all the science news I got. But yeah, if anyone else has any kind of like science-y thing to talk about or whatever, you'd probably just like tell me it or message me it, and then we can, during the next science stream, we can look into that. But I figure there's not really a reason to start a game or anything at this point, since it's already been about over an hour and a half. But I thank you to everybody that came by today. I hope you had a good time learning some science and learned some new things that have been happening. And I hope you all have a good rest of your day or night, and I will see you next time.